Today's is Professor Will, CBMD, PhD. Today's Thursday, July 18, 2019. It is 5:08, 42 seconds Eastern Standard Time. And this is a continuation of my scientific research on microbiology. So let's hop to it here. Protein secretion, or protein export, is the movement of proteins from one cellular compartment to another, or to the exterior of the cell. In bacteria, this involves the movement of pre-secretory and membrane proteins from the cytoplasm, their initial site of synthesis, to a number of aqueous or membranous compartments, depending on the type of bacterium under study. In recent years, it has become evident that a variety of pathogenic bacteria are also capable of secreting their proteins into animal and plant cells. Bacteria possess several different pathways for promoting protein secretion, and these are generally classified according to the secretion machinery utilized for protein export. The most commonly utilized pathway is the sec-dependent or general secretion pathway, GSP. The GSP promotes protein translocation across the cytoplasmic membrane. It is driven by a collection of cytoplasmic peripheral and integral membrane proteins that collectively known as the sec machinery of translocation. The GSP can be thought of as the major housekeeping protein secretory pathway of the bacterial cell. It is homologous to the pathway promoting import of pre-secretory and membrane proteins into the endoplasmic reticulum of eukaryotic cells. Certain gram-negative bacteria possess terminal branches of the GSP for secretion of proteins across the outer membrane. Besides the GSP, there are several other specialized protein secretion pathways that are utilized by more limited sets of proteins. These include I, the twin arginine protein secretion pathway that promotes the export of redox cofactor-containing enzyme, II, ABC protein secretion pathways that are driven by ABC, ATP-binding cassette exporters, and promote the secretion of toxins, proteases, lipases, and specific peptides. III, the type 3 or contact-dependent protein secretion pathway that promote binding and import of proteins into animal and plant cells. The latter two pathways are typically associated with bacteria pathogens and specialized microbial niches. General secretion pathway, targeting signals on preproteins. The hallmark of GSP-dependent pre-secretory protein, preprotein, is the possession of characteristic signal peptide that is often located at its amino terminus. Such signal peptide serves as recognition elements for the sec machinery, and they also delay the rate of protein folding, a potentially important feature since protein export by the GSP must occur prior to the completion of protein folding. Secretory preproteins can interact with the sec machinery in either a co-translational or post-translational fashion. Sec-dependent signals, peptides range in length from 18 to 30 amino acid residues, typically being longer in gram-positive bacteria. Although they possess little in the way of primary sequence homology, they do contain three characteristic regions. 1. A short positively charged N domain. 2. A hydrophobic H domain of 7 to 15 uncharged amino acid residues. And 3. A short polar C domain that contains a signal peptidase cleavage site. Two consensus cleavage motifs exist within the C domain, one for general secretory proteins and one for lipoproteins. Lipoprotein need to be modified with diglyceride on the cysteine residue at the beginning of the mature region to allow signal peptide cleavage. Mutational studies show that the H domain is the most critical for signal peptide function, and truncation or introduction of charged residues within the H domain often prevents export of the protein. In contrast, the N domain appears less critical, although its overall charge can affect the kinetics of protein secretion. Alteration of the C domain may prevent signal peptide cleavage, while the resulting preprotein is still secreted. It remains tethered to the cytoplasmic membrane by its uncleaved signal peptide. The signal peptide is thought to function as a loop, allowing insertion of an amino terminal hairpin of the preprotein into the cytoplasmic membrane. Whether such insertion occurs directly into the lipid biolayer by analogy to model systems, or is promoted by prior interaction with the translocon is unknown. Additional recognition elements are contained within preproteins, since signal peptide deletion still allows a low level of protein secretion to occur. Residual export can be greatly augmented by certain mutations in the sec machinery that generally suppress signal peptide defects. One type of recognition element for chaperones that targets preproteins to the translocon appears to be exposed hydrophobic protein surfaces. It is thought that delayed folding kinetics of preproteins with resultant hydrophobic surface exposure facilitates preprotein interaction with specific chaperones for maintenance of the export competent state. However, the structural basis for such recognition has remained largely illusory. The final sorting of secretory proteins to the periplasm cell wall and other membrane or protein layer occurs subsequent to translocation across the cytoplasmic membrane. The paraplasm is probably a default pathway that requires no specific sorting signals to achieve proper targeting. By contrast, topogenic signals are required to promote association with the cell wall or outer membrane. The nature of these signals is only now being clarified for specific systems, and it is unclear at present how general such signals are. For outer membrane protein, targeting the possession of B structure or ability to bind lipopolysaccharide appears to be important. In E. coli, lipoprotein is the identity of the second mature. Transmembrane segments serve to initiate membrane protein insertion, while even numbered segments serve as membrane anchor domains to arrest translocation. Insertion of an amino terminal portion of a membrane protein in an N-out, C-in orientation, so-called amino terminal tail insertion, has been shown to be independent of the GSP and may require a novel pathway promoted by a homologue of OXALP, a protein responsible for amino terminal tail insertion of mitochondrial membrane proteins. Chaperones and targeting factors. As nascent or newly completed chains of presecretory and membrane protein emerge from the ribosome, they encounter a variety of chaperones that maintain them in an unfolded state and target them to the translocon. While a number of chaperones have been shown to play at least a minor role in protein export, I-trigger factor, a ribosome associated with proline isomerase, and heat shock proteins GROE, DNAK, DNAJ, and GRPE, two major chaperones have been characterized in E. coli. Sec-B, a signal recognition particle SRP. Sec-B is present in gram-negative bacteria. Its existence in gram-positive bacteria is uncertain, while SRP is conserved among all three domains. Sec-B is a small tetrameric protein that binds a particular group of preproteins, either co-translationally or post-translationally, and targets them to the translocase through its specific interaction with Sec-A, a subunit of translocase. Sec-B is non-essential for cell viability, although its absence results in substantial secretion defects for Sec-B-dependent protein. Unlike the more complex heat shock chaperone, Sec-B is not an ATPase, and it can only prevent protein folding, not reverse it. The structural basis of Sec-B preprotein recognition is unclear, although it requires a hydrophobic patch on Sec-B. Of interest, this region overlaps with acidic residues that are Sec-A binding site on Sec-B. These results have led to a model where preproteins are passed from Sec-B to Sec-A by a handoff mechanism. Bacteria possess an SRP particle and SRP receptor that is homologous to its eukaryotic counterparts and are essential for cell viability. While there is no evidence for a protein translation arrest by bacterial SRP, the membrane targeting function of bacterial SRP appears to occur similarly to eukaryotic SRP. The interaction of FFH with proteins must occur when they are still ribosome associated, and it is dependent on the hydrophobicity of the signal peptide or signal anchor domain. While characterization of E. coli SRP receptor has proceeded well, considerable controversy surrounds the identity of the in vivo substrates of SRP. While SRP mutation or depletion affects somewhat the secretion of certain paraplasmic and outer membrane proteins, recent data indicates that this system plays an important role in the biogenesis of particular integral membrane proteins. The effects of SRP depletion on paraplasmic and outer membrane secretion may be indirect, since Sec-Y protein requires SRP receptor and presumably SRP for its biogenesis. Good day, this is Professor Will, CBMD PhD. Today is Thursday, July 18, 2019, at 6:36, 11 seconds Eastern Standard Time. And this concludes my reading on general secretion pathway for microbiology. Hope you enjoyed listening. Have a great day.